However, guys, I know we ended the previous few slides on a bit of a downer, but it's not all over. There are many organisations supported by academic studies that are intent on trying to save what we can and scale back our impact on the planet. Now, you can always research the case studies we've discussed or find your own and look at what organisations are trying to do to stop further decline. There are lots of good websites for this, such as the WWF and the Nature Conservancy, but there's also the Woodland Trust, and there are many, many, many more. I can't fit them all on here, or begin to even look at the ones that are all over the world, so do please take a look at those. So, bioinformatics and conservation biology, you wouldn't have thought the two really go together. And bioinformatics is the application of computational tools to analyse biological data, so genetic sequence or protein sequence. It is a very new tool to conservation biology. So, bioinformatics helps conservation in a number of ways, but the two main ways are providing freely available databases to store genetic information, and this is very important. This is where I accessed all of the genetic data that I have used for all of the modules as part of this website so far, because after people have conducted studies, they always deposit their genetic data into a database which is freely accessible by anybody for anybody to analyse. So bioinformatics can also help by analysing that genetic sequence to reveal information such as evolutionary history and what taxonomic family the organisms belong to. We will cover that in more detail in the practical lecture for those of you who aren't quite entirely sure about taxonomy and phylogenetics, and those of you who've done the evolution module probably won't need to review it, but if you do feel unsure, that's something you can review. But why is any of that important for conservation biology? Well, essentially, we need to decide who to save, and this is not an easy question and there is no right answer. There are many different approaches to this question, and there is much debate in the scientific community. Conservation projects cost a lot of money and there's only so much funding available. And conservation approaches are very complex. One of the most classic ways to view this is the preservation of biodiversity, and this is the species-based approach. Now, the species-based approach is to essentially preserve as many species as possible. Now, they could be threatened, ecologically important, with economic value, or with cultural value. And one of the quotes I found, I don't know who it's from, so if you do know, do let me know, which says, "Save the last of the least and the best of the rest," and that pretty much sums up the species-based approach. However, it's not the only approach. There are lots of different approaches, and for valid reasons, some scientists do indeed disagree. So if you get some time, do feel free to research other approaches and see if you agree. And then you can discuss it in the forum if you do so feel that would be useful. So back to bioinformatics. So bioinformatics can help decide what a species is. It can do this through the construction of a phylogenetic tree. This has also been named phylogenetic diversity. So the construction of a phylogenetic tree is often done in conjunction with ecological studies, such as population geography, to assess gene flow. So can the two populations interbreed? This has important conservational consequences. If there is some debate as to whether or not two populations are members of the same species or not, we could analyse their DNA to find out. If they are found to be two different species, they will likely get separate conservation statuses, which may impact future conservation efforts. So we'll take a look at a case study. So originally it was thought that the sandwich tern, which is a great name, comprised of one species and two subspecies. However, observations revealed minor differences in morphology and important differences in breeding habits. So an earlier study had looked at the relationship between the different birds in the tern family, but the sandwich tern group was still largely unresolved. So the study question was, is there a current phylogenetic understanding for this group? Is it correct? Are they all one species or are they multiple? So they collected DNA via blood samples and constructed a phylogenetic tree, and the results came back that the group actually comprised of two species. So what was the outcome? So the name changed for the two species to reflect that they were indeed separate species, and the authors recommended a revision in the IUCN listing and a conservation assessment on the newfound species. So you can see how that is important for the future conservation efforts. Now the next case study is on an Ethiopian warrior ibex. So the ibex resides along with a similar looking ibex called the Nubian ibex in Ethiopia. However, the warrior ibex inhabits only a small range in the Simon Mountains. It is already classed as critically endangered due to illegal hunting, habitat loss, climate change, disease and competition with livestock. Now there is only one population left, which I'm sure if you think about your genetic diversity, that's not very future-proofed at all. Although it was already classed as a distinct species, the previous classification was done primarily on morphology, and there was some opinion that perhaps the warrior ibex is likely a subspecies of the Nubian ibex, which would mean it wouldn't necessarily need such rigorous conservation efforts because it would just be a subspecies of another ibex whose population is perhaps not as suffering as much. So the study question was, is the Ethiopian warrior ibex a single species or is it a subspecies of the Nubian ibex? So They collected DNA from fecal pellets and constructed a phylogenetic tree. They also used population genetics and ecological data in the study. So the results were back. The Walia ibex is confirmed as a single species with very low genetic variation and one population of about 450 animals, which is very low. Going back to our population genetics slides, why is having a low genetic variation and a single population likely to be a problem for the future of the Walia ibex? I'll leave that up to you to think about. And what was the outcome? So the authors called for urgent conservation actions such as the reintroduction into their historic range. So if you do feel like looking up anything, you can always follow up on the Ethiopian Walia ibex and did the conservation efforts work? Have they indeed reintroduced their range? Has anything been followed up after the study? I'll leave that up to you to research if it's something that interests you. Now the next one is a little bit of a weird one. I thought I'd do it just because it's a little bit different. So the caterpillar fungus, which is this critter up here, is one of the most valuable medicinal fungi on the planet. It is endemic to the Tibetan Plateau. It is used in traditional Chinese medicine to treat a wide range of conditions including asthma and lung inflammation. So trade was legalised in 2001 in Nepal, and since then the local market price has risen 2,300%, but harvests have declined. The caterpillar fungus is harvested after it has infected, killed and mummified the larva of a moth, and then produced a fruiting body which is part of the medicine, which is pleasant. This is the larva body of the moth that's unfortunately mummified, and this is the fruiting body of the fungus, which is the medicinal bit. It is now classed as an endangered species in China, and this is thought to be due to overharvesting. Now there was some queries to the taxonomy of this fungus. A previous study using morphological characteristics has identified 5 potentially new species related to the caterpillar fungus. The Tibetan Plateau is a highly mountainous area and thought likely that gene flow had halted between some populations. So the study question was, what is the genetic diversity and evolutionary history of the caterpillar fungus? So they collected specimens and extracted DNA. They then conducted population genetic studies and a phylogenetic tree. Although these fungi are spread over a large area geographically, it appears in fact that they are just one species. Genetic population studies revealed that the southern isolate had more genetic diversity than the northern ones. So the outcome of this study was that the authors recommended that the southern, more genetically diverse populations be recognised as important genetic reservoirs. Due to the economical and medicinal properties of the species, they recommended the species be considered for conservation efforts. So that's it, guys, you've made it. You've made it through that long-winded, very complex PowerPoint on biodiversity. So please feel free to take a look around the extra resources section I've compiled. I think they might actually be very useful. So I highly recommend going to the California Academy of Natural Sciences. I've linked to them in the extra resources section. They have about 35 short videos covering many aspects of biodiversity. That includes topics we've already covered and those that we haven't, but importantly they're slightly different. It's not like they're reading off of another PowerPoint. They've got videos and animations to show and illustrate their points, so they really are very interesting and useful. So I recommend you give them a go. Once you're ready, head on over to the practical exercises and we can get to building our phylogenetic tree and look between the relationships of some of the big cats. So we're going to be taking a look at how bioinformatics help shape conservation efforts for the 2 endangered clouded leopard species. So take a break, guys, have a look at the extra resources, and then when you're ready I'll see you over the practical section.